Over my hoarse voice here. It's my pleasure to welcome Dr. Kabir Peay to the EEB seminar series. Thank you for enduring an onslaught of seminars this week, but this one will be great too, I promise. Kabir originally is from the Bay Area, but has moved around a bunch. Did his undergrad at UC Santa Barbara, and then a master's at the Yale School of Forestry, and then he did his PhD at UC Berkeley with Tom Bruns and Matteo Garbaletto.
Good memory.
Where we met as grad students. And from there, he was a postdoc at Stanford for a little while, then took a faculty job at Minnesota, and was quickly snatched back by Stanford. He decided they wanted him actually as a faculty member. So he's been there for a number of years. He's now a professor there. And Kabir works on interactions between fungi and plants. And his work spans a number of different scales. So he thinks some about the fine scale interactions at the tips of roots, all the way up to broader questions about community assembly and turnover of species across biogeographic gradients. So a really broad community ecology thinker who happens to work on fungi. So thank you very much, Kabir.
Yeah, thanks, Nathan. And thanks for the invitation to come. I'm sort of embarrassed that this is my first visit to UCLA, but I'm happy to have rectified that problem now. You guys have a beautiful campus and a nice community of scientists here. So can everybody hear me? I'll try and speak up if I'm a little bit quiet. So what I want to talk to you today about is some of the work that I've done in these fungal communities that Nathan has been describing. And I realize you guys are inundated with talks on microbial ecology. And so you are probably more aware than anyone that microbial ecology has been a real flourishing area of the broader field of ecological research. And when I say microbes, I use this term fairly broadly. Some people have size cutoffs or whatever, but I use the term microbe just to indicate things that are studied by microbiologists using microbiological techniques, things like bacteria, viruses, and fungi. And in fact, in the time that I've been studying ecology, one of the biggest changes I've seen, at least at the Ecological Society of America meetings, is the number of talks that have been dedicated to microbes went from one at my first ESA in 2004 to multiple concurrent seminars in sort of current ESAs. And one of the reasons that I think there's been such an explosion of research and interest in microbial ecology has been what I see as a sort of positive feedback between different disciplines. So on one hand, I think ecologists have more and more gotten access to advanced molecular techniques that enable people to sequence large amounts of DNA and identify microbial organisms. And microbiologists are increasingly starting to use ecological theories to try and understand the systems that they're working in. And as a result of this positive feedback, I would say that we're in something of a new era of discovery. This is sort of similar to when people like Humboldt or Darwin or Wallace were getting on boats and sailing off to new parts of the globe and trying to understand the patterns of the flora and fauna that they saw. We're sort of also in this golden age of discovery in microbial ecology. So we've done incredible things now, like mapped the human microbiome. This is a figure here showing the different kinds of communities of bacteria that live on different parts of your body. And you get people doing crazy studies like this one, where they sort of map out different quadrants of their face and look at the biogeography of microbes on things like your teeth to your face. And as a result of this kind of sequencing of the environment, we're getting more and more examples of really close, intimate interactions between organisms. So we're getting this feeling that we and other organisms are awash in the sea of microbes. And often we form these really intimate symbiotic interactions with these different organisms. So here I've I've shown you a figure that I really like from a study that shows actually a symbiosis between different microbes. And what you see here, these kind of long tubes, are the hyphae of a fungus. And this inset here shows fluorescent labeling of bacteria that live inside the hypha of this fungus. And they produce a toxin called rhizoxin, which allows this fungus to cause disease in rice plants. So without these bacteria, the fungus can't do it. And as a result of the sort of accumulation of these kinds of examples, I think we're undergoing somewhat of a paradigm shift in the way that we think about interactions with microbial organisms. So I think in the past, we've often thought of microbes in a very antagonistic sense, sort of colored by things like the germ theory of disease. And you can see this sort of spirit exemplified in this quote from Schwendener, who is the German that first described the lichen symbiosis. And you can see that when he described the symbiosis, he used these really pejorative terms. He thought that the sort of fungus was a master and a parasite, and that these algae were slaves to this fungus here. But as a result of a lot of this sequencing and sort of accumulation of these good examples of symbiosis, the paradigm has shifted and we now recognize that positive symbiotic interactions are equally common in nature. So despite the fact that we recognize the prevalence of these kinds of positive symbiotic interactions, I would say there's still a lot of really basic ecology and evolution that we don't understand about even some of the most common forms of symbiosis. So with that lead-in, I work on a specific type of symbiosis between plants and fungi that's known as mycorrhizal symbiosis. And mycorrhizal literally means fungus root. And it refers to the fact that these fungi, you can see all these white threads here, these are fungi, and they're growing into the roots of this plant. And they form this new absorptive organ known as a mycorrhiza. And this is, I think you could make a case that this is one of the most pervasive forms of mutualism in terrestrial ecosystems. It's pretty ancient, so it goes back to about 500 million years, so really when plants were first colonizing the land. And this is the ancestral state for the vast majority of plant species. And so I think that it's been estimated that 90% of plant species form some type of mycorrhizal symbiosis. So it's more unique to be non-mycorrhizal than it is to be mycorrhizal. And you could think of this really as a biological marketplace where plants and fungi are exchanging limiting resources. So usually from the plant side, this is photosynthetically fixed carbons that are traded down to the fungus in exchange for nutrients like nitrogen and phosphorus that the fungi scavenge from the soils. And this can account for fairly large fluxes within ecosystems. So in some places it's been estimated that up to 90% of plant nitrogen can come through these symbioses. And fungi often get on the order of 10 to 15% of plants' net primary productivity. And so if this is, you can imagine if this is something you spent 15% of your net income on, this is something that you value quite a bit. And it has big consequences. This is the obligate big plant, little plant. You can see this plant is mycorrhizal and this plant is non-mycorrhizal. And there's quite a big difference in the size and performance of these two different plants. One of the really interesting things about mycorrhizal symbiosis is that, whoops, this thing jumped ahead really. Oh. Okay. One of the really interesting things about mycorrhizal symbiosis is that, whoops. Sorry, I think I missed something here. There we go. Is that there are many different forms and lineages of mycorrhizal symbiosis. There's not just one kind. And this involves many different lineages of both independent lineages of fungi, independent lineages of plants, and many, many species within each of these different types of associations. What I'm showing here is the two predominant forms of mycorrhizal symbiosis. On your right, this is arbuscular mycorrhizal symbiosis, or AM, as I'll abbreviate it. And on the left here, this is ectomycorrhizal symbiosis, or ECM, as I'll abbreviate it. These are the two most common forms of mycorrhizal symbiosis for plants. And they have very different anatomical features. These ones grow on the outside of roots and kind of around the cortical cells of the root. These things actually penetrate the plant cell wall. And they have different reproductive morphologies. So a lot of the big terrestrial mushrooms you see are the product of ectomycorrhizal fungi, whereas these guys really make kind of big spores only rather than fruiting bodies. Let's see. Most of my research is focused on this particular form of symbiosis, ectomycorrhizal symbiosis. And it's suspected that given the evolutionary history of these different lineages, that these two different types of mycorrhizal fungi have pretty different physiological capabilities. And so their presence or their prevalence in ecosystems can actually have pretty big consequences on the way these ecosystems function. There's been a nice study recently that suggested that systems that are dominated by ectomycorrhizal fungi tend to store more carbon per unit nitrogen. And then there was a really nice paper that just came out quite recently that showed that the strength of conspecific negative density dependence, which is something that people think is really important in controlling the diversity and distribution of plant species, that this actually varies fundamentally between plants that have ectomycorrhizal versus arbuscular mycorrhizal symbiosis. So these are probably fairly important interactions. One thing, though, that I will point out about all these studies is even though they show these symbioses in general can be very important, a lot of them are done in the absence of any information really about the community of these organisms themselves. And so the communities of fungi that are involved in these symbioses are really black boxed in these studies. And so that's something that we really want to try and get into in our lab. And so in our lab, we really try and explicitly unpack, using a lot of these molecular tools, the structure of microbial communities, and in particular these ectomycorrhizal communities. You can see here a little root that's covered with hyphae here on the outside. And we try and ask questions about how these things interact with both plant communities and then ecosystem processes. So all this kind of dead debris here is meant to indicate decomposition. And we ask really two kind of big questions. One is, why is it and how is it that ectomycorrhizal fungal communities change over space and across environments? And then the second question is, how do these changes influence other things like plant communities or ecosystem processes? So for today's talk, what I really want to do is take you to an ecosystem that I've worked in quite a bit. This is Point Reyes National Seashore. And this is a little bit faded, but you can see there's some interesting patterns in the vegetation that you observe across this landscape. And what I want to try and get you to do is imagine this landscape from the perspective of the microbial organisms that live in there, and then try to understand how it is that these microbial organisms themselves actually shape the patterns that we see across this landscape. So the way in which I'm going to do this and structure my talk is through talking about work that we've done at different spatial scales in particular. The importance of scale is something that ecologists have recognized for a long time. This recognition that particular processes operate at spatial and temporal scales that are often disconnected from the scale at which we do ecological studies. And because of that, it's important to be aware of these other larger scale processes and account for them in our studies. And it's interesting to think about the importance of scale from the perspective of microbial organisms, because for historical reasons, people studying microbes have really only focused on these sort of local scale interactions. And I'll get into why that is in a second. And I should just say, sort of in this diagram here, you can see we go from kind of local, smaller spatial scales to larger spatial and temporal scales. And I've tried to outline some of the key kind of drivers of community composition and structure that might operate at these different scales. So for example, at local scales, we expect that the environmental filtering and things like competition are going to be really important in determining who's in a particular place. While as we go up to larger and larger spatial scales, things like the evolutionary history of a particular region is going to be important in determining what the regional pool of species are. All right, and so for my work, what I want to try and do is talk to you first, starting at the landscape scale, about some work that I've tried to do to try and determine whether or not larger scale processes that operate across the landscape are important in structuring communities of these ectomycorrhizal fungi. All right. So just to give you some background as to why I think it is that these kinds of research, this kind of research on larger scale processes and their effect on microbial communities has been somewhat overlooked until recently, is that traditionally people have thought that lower organisms, so bacteria, fungi, lichens, algae, people thought that they had no limits to their dispersal. And you can see this exemplified here in this quote from an eminent British mycologist named Miles Joseph Barclay. And he says that indeed, were fungi not so much the creatures of peculiar atmospheric conditions, there would seem to be no limit to the diffusion of their species. And this is in fact a letter he was writing to Charles Darwin. And so you've heard there are some modern incarnations of this kind of idea. The Bass-Becking hypothesis, everything is everywhere but the environment selects, is something that has been somewhat prominent in the literature recently. And so a corollary of this idea that microbes have no limits to their dispersal is this idea that microbes tend to have really high levels of alpha diversity, but very low levels of beta or gamma diversity. So there's fairly little, you can get lots of species in a small area, but as you go to larger and larger areas, you tend to detect fairly few new species. And this is sort of illustrated by this quote here by G.R. Bisbee. And he says there are perhaps three times as many phanerogams, so plants, as fungi on Earth. Nevertheless, species of fungi can be expected to outnumber species of plants in any particular state or country. Okay. So I wanted to try and test some of these traditional assumptions about the structure of microbial communities. And the way that I wanted to do this was through the framework of island biogeography. So this is, of course, the brainchild of E.O. Wilson and Robert MacArthur. But since we're here, I will say that Martin Cody's book, Plants on Islands, was actually one of my favorite books as a graduate student, and it was really helpful for me in thinking about some of these projects. And the way I tried to approach this was as such. So under a sort of classic kind of island biogeography theory, there's expected to be a relationship between the number of species on a particular island and the geography of that island. In particular, as island area increases, I've shown you a little cartoon where the kind of classic expectation is as you get larger islands, you get larger populations of species that are less susceptible to extinction. Extinction rates go down, and you have a greater diversity of organisms on these larger islands. By contrast, I just showed you this sort of traditional microbial idea that microbes, they can have incredibly large populations in very small areas, less subject to this kind of stochastic extinction, and we'd expect that they have very high levels of diversity even in small areas, but very little increase in diversity as you go to larger and larger areas. All right. Sort of the second hypothesis you might have is that based on kind of traditional island biogeography expectations, as you go to islands that are more and more remote from a mainland that is a potential source of new colonists, there's an expectation that there's a decrease in the sort of dispersal of organisms to those islands that leads to a decrease in colonization and a decrease in richness on islands as you go to more and more isolated islands. So then you can contrast this with this kind of traditional microbial view where you would think that really there's no limits on dispersal, so dispersal is kind of unlimiting, and even as you go to more and more remote islands, you would expect to see really no decrease in richness of these organisms, microbial organisms on more and more isolated islands. We can contrast these kind of traditional microbial with our kind of standard expectations from island biogeography. So the system I worked in is this one here. This is, as I said, Point Reyes National Seashore, which is about an hour and a half north of San Francisco. And these patches in green that you can kind of barely make out here, these are patches of a pine species called Pinus muricata. This is bishop pine. And this is sort of an interesting coastal pine. It's got serotinous cones that really only open after stand replacing fires. And what I'm showing you here is a GIS map that shows a large fire, the Vision Fire, that burned through a big section of the park in 1995. And this green shows the pre-fire distribution of this bishop pine. And after the fire, there was quite a lot of recruitment of this pine out into these areas, previously uncolonized areas of coastal scrub vegetation. So in the days before Google Earth, what I was able to do is get high-resolution aerial photographs and begin to map the distribution of these bishop pine as they encroached into this coastal scrub area. So we're going to zoom in here. You can see this is a Limantour parking lot. If you've ever been to Point Reyes, there's the beach. We're going to zoom in on this little spit of land right here. And as you go in, you can see these little green dots. And each of these individual green dots is an individual pine tree. So you can map the distribution of these pines in this landscape at a fairly fine spatial scale. And here, this is that same pine, individual pine right there. So the approach that I took was to treat these pines as islands. And this is sort of an approach that I stole from Dan Jansen, who proposed that you could think of host plants as islands in ecological or evolutionary time from the perspective of the organisms that use them as resources. And so this works in this system because Pinus muricata is really the only ectomycorrhizal host plant that's common out in this coastal scrub area. And it's surrounded in this matrix of non-host arbuscular mycorrhizal vegetation. And so since these things are not suitable to these ectomycorrhizal fungi, they can't colonize this vegetation, these really are like islands in a little sea of non-host vegetation. There's some other nice advantages about this system. Because the pines recruit after stand replacing fires, you can control for the successional status of the vegetation somewhat. And the soils in this particular part of the park are relatively homogeneous. There's not any large environmental gradients we have to worry about. And so what we really get is these islands of pines that vary in the things that we're interested in, the size of the islands and their relative isolation. Okay? And so we can test some of these expectations from island biogeography on these pine islands. So what I did is went out and found a bunch of these patches, mapped their area, and randomly selected a small subsample of islands to go out and sample the ectomycorrhizal fungi on these pine islands from. We went from very large, sort of about a hectare in size, down to quite small, so single individual pines. So to look at area, I had to use a slightly different part of the park to try and get a larger isolation gradient. And what I did was hold the sort of pine island area constant. So I sampled single individual trees going from the edge of this main body of pines right here. And this is what I will refer to in some of these studies as the mainland. So this is kind of our mainland model. And going out from individual trees right from the edge of the forest up to over a kilometer away from the edge of the forest. And on each of those patches, I use molecular tools to try and identify the fungi that colonize the roots of these different pine islands. And just to sort of show you what that process looks like, this is my pine island, and this is a little bit of dirt. So I'd go out and take soil cores, and from those cores, I'd extract the roots of these plants and identify individually colonized ectomycorrhizal roots. You can then extract the DNA from those individual roots and then use fungal-specific primers to amplify genes from the fungal internal transcribed spacer region, which is kind of a fungal barcode that we tend to use. We used what is now sort of an old technology. This is Sanger sequencing, so you can generate individual sequences using this. And those sequences then can be clustered into operational taxonomic units, or OTUs. In the case of fungi, the particular locus that we use is fairly fine scale, and so these are basically species, all right? Unlike bacterial 16S OTUs. And then we can take these sequences and search against online databases to identify known taxa, that are on these roots. So just to give you a sense for what we found, this is the kind of first result. And what you can see is that this is ectomycorrhizal species richness on the y-axis. And then this is the area of these individual pine patches. And you can see there's a fairly strong positive relationship between the size of these pine islands and the number of species that colonized ectomycorrhizal species that I found on these islands. And this is still probably the best R-squared that I've had as a research scientist, so I quite like this result. As I'll note, this isn't a log-log scale. This is often how people plot these things. And the convenience here is that the slope of this relationship is referred to the z-value, and it gives you some sense for how steep or shallow this line is. And you can use the z-value to compare across different studies. And so just to put the value that we get there, the z-value of 0.2 in perspective, this is the first study that really came out looking at taxa-area relationships for microbes. In fact, it was a pair of studies on bacteria and fungi that were both published in Nature. And you can see these z-values for these two different taxa were quite low, more consistent with this traditional view of microbes as having very high alpha but very low beta or gamma diversity. Whereas the z-value that I got for this particular study is around 0.20. And this is much more similar to what you would see for macroorganisms. So still, there have been fairly few studies on species-area relationships in microbes, in part because they're actually kind of hard to do. If anyone's curious, I can talk more about why I think my results are robust or why I think they might be different from these particular studies right here. But what I will say is it kind of behooves us, particularly in a rapidly changing and fragmenting world, to figure out if microbial diversity looks like this or if it looks like this, because it's going to have big consequences for conserving the diversity of the microbial organisms in these habitats that we're destroying. So if we go back to this kind of traditional, this dichotomy that I put up here between this kind of traditional microbial view of the world versus this kind of standard island biogeography type view, at least in this particular system, we kind of reject this traditional microbial view of biodiversity. So going on to the second part of this particular study, when we looked at the richness of ectomycorrhizal fungi along an isolation gradient. This is, again, richness of the ectomycorrhizal fungi on these individual tree islands, plotted against the distance of these individual tree islands away from the forest edge, so away from the mainland. And what you can see, again, is that there's a fairly significant decrease in richness as you move away from the forest edge. It translates to about a 50% decrease in species richness from over a distance of about 1 kilometer. All right. Whether or not this is significant from the perspective of the plants is something that's interesting to think about. And also something that to me is quite interesting is because these are individual trees, you can see that individual trees are often associating with at least a few dozen species of ectomycorrhizal fungi. What the functional consequences of this are, or how this kind of diversity is maintained in these systems is actually quite interesting and relatively unexplored. So again, if we go back to these two paradigms that I put up here, this kind of traditional microbial view of the world where microbes are dispersal unlimited, and then this sort of standard island biogeography view where you'd expect decreases in richness with increasing isolation. Again, we kind of reject this traditional microbial view of the world here. So one thing that I will say is that both of these results are patterns that are consistent with an island biogeography model, but they're not direct tests of the mechanisms that are thought to underlie island biogeography theory, things like extinction and dispersal limitation. So what I wanted to try and do after these two studies was follow up and try and get a little bit more mechanistic and see if I could test directly whether or not dispersal limitation, at least, was in some ways contributing to some of the patterns we saw with respect to isolation. So to do this, I took an experimental approach. I created these little experimental islands, these little sampling stations here, and I put these out in a matrix across the landscape. Again, these are embedded in this arbuscular mycorrhizal vegetation, and I put them in places where I knew where the closest host trees are. And again, because these host trees are the only potential source of propagules, I knew how far away they were from any particular source of fungal spores. And then in each of these sampling stations, I put pine seedlings, and these are growing in sterile soil, and so the seedlings are initially uncolonized. You can see they're not in contact with the ground, and so the only way they can be colonized is from aerial spore inputs. I also had these really fancy devices right here. This is a funnel stuck into a mason jar. This is what I call a spore trap. People will sell you these things for like $15,000, but you can do it at Home Depot for cheaper. And so in these things, I basically just caught any sort of dry deposition or wet deposition that I could at each of these sites, and then used some molecular techniques to directly measure the quantity of spores and the diversity of spores that landed at each of these sites. So we use two molecular techniques to do this. The first is quantitative PCR. This basically lets you count the absolute number of spores in a particular sample. This is a little bit more restrictive in that you need to design specific assays for each species you want to measure, so you can only do it for a couple different species. So we designed qPCR assays for 3 different species that we knew were quite common. And then to look at spore diversity, we actually used one of the first next-generation sequencing tools that came out. This is 454 Pyra sequencing. This tool is obsolete now, so nobody uses it anymore. But at one time, it was super popular. And it was so popular that there was a hip-hop band in Oakland that actually called themselves 454. And there were questions at the time about how good this data was going to be. People had questions about homopolymers and things. But you can see these guys were actually strong believers in the quality of the data that you get from this type of experiment. So this is real. I didn't actually make it up. I just saw this on a billboard driving home one time and was amazed. Okay, so just to give you an overview of the landscape we were working in, this is again Point Reyes, and you can see these yellow pins just show you each of these individual kind of experimental stations that I set up across this landscape and across this isolation gradient, going away from these established pines. So what we found was actually fairly clear cut. This is the qPCR data for the 3 most common species we found colonizing these seedlings of bishop pine. And you can see in each of these I've plotted spore deposition rates and sort of spores per square centimeter per day, plotted against distance away from the forest edge, so distance from this mainland. And what you can see is that for all 3 taxa here, there's a pretty rapid exponential decrease in the number of spores that reach these stations as you move farther from the forest edge. Again, since this is log-log, this is a much actual steeper decrease than you would see from just the linear relationship here. There are some differences here between taxa, so the slope of these lines are actually similar between all these different taxa. But what you can see is there's actually some difference in the intercept. So this fungus here, Solus pungens, is sort of the champion spore producer in this ecosystem, whereas the other guys tend to produce much smaller overall quantities of spores. All right. We also found that the spore richness of ectomycorrhizal fungi decreased pretty quickly as you moved away from the forest edge. So this is the number of ectomycorrhizal spores we got in each one of these spore sampling traps. And I forgot to mention before that the black and white are just the 2 years that I repeated this study for, so I did it twice. And you can see again there's a pretty significant decrease in the richness of ectomycorrhizal spores reaching these stations as you move out a few kilometers away from the edge of the forest. I'm not showing you the seedling data, but the seedlings actually showed identical patterns in terms of the richness of fungi colonizing individual seedlings as you moved away from the forest edge. All right. So again, pretty consistent evidence that there's a strong dispersal limitation operating in the system. So just to summarize this particular portion of the research at the landscape scale, what I've shown you is that there's quite a bit of landscape scale heterogeneity in the diversity and composition of these ectomycorrhizal fungi, and that it seems like this is related to habitat size and isolation. And we've shown some direct evidence that some of the processes like dispersal limitation are driving these landscape scale patterns. And this is what I would say important in a way that people hadn't previously suspected for microbial organisms. So because we know that this sort of landscape scale, these landscape scale processes, and what I mean when I say landscape scale, these are things you can't actually measure at the individual sample scale, right? You can measure local environmental conditions, but you can't measure at the local scale how isolated something is or how large the patch it is in. And those kind of landscape scale processes seem to influence the diversity of these ectomycorrhizal fungi. But because we know that there's these landscape scale influences on local community structure, what I really wanted to do next was ask whether or not the changes that we see across this landscape in the local structure of these ectomycorrhizal communities, whether or not this actually influenced other local scale interactions. And what I mean by this in particular is interactions like competition between different plant species. All right? So does the kind of variability that we see actually have some consequence for other organisms? All right. And so I have some evidence from the previous study I just showed you that suggests that there should be some important physiological consequences for the plants based on this kind of heterogeneity. So this just shows you the fraction of the root systems of these experimental pine seedlings I had put out that were colonized by ectomycorrhizal fungi across this isolation gradient. And you can see that as you move farther away from the forest edge, these plants, the colonization intensity goes down pretty significantly. And just from this figure right here, these are two seedlings that were in the study. They're both the same age. This one was just never colonized by ectomycorrhizal fungi. It came from an isolated site. This one was from a forest edge site. It was highly colonized and was quite large. So there's some suspicion that there should be important physiological consequences for plants. But to me, just this data alone kind of ignored two things that I know are important and that I wanted to try and address. The first is that this data ignores any kind of temporal component to the colonization process. And what I mean by this is that over time, many of the seedlings that were in my study probably would have been colonized if they'd been left out for 2 years or 3 years. Eventually, spores would have found them. So there's a temporal dimension here. And in addition, some of these seedlings that were minimally colonized, they should eventually be fully colonized by ectomycorrhizal fungi through vegetative spread of the hyphae of these organisms. They're vegetative, and so they can kind of clonally grow throughout the root system. So that kind of was ignored in this sort of temporal dimension in this particular study. The other thing is that when we do these kind of big plant, little plant comparisons that people often do when they look at the advantages of being mycorrhizal versus non-mycorrhizal, what it ignores is that in real environments, plants actually have to establish in competition with other organisms that are around them. And so whether or not this kind of a biomass difference is actually important is going to depend a lot on who is around and how the plants are competing with other species they might coexist with. So to try and account for those two factors, I did a fairly simple growth chamber experiment. And so I just went out and dug up soil from the coastal scrub in two areas, one that was close to where these established pines were, and one that was very far from where these established pines were. And I set up a bioassay experiment. So I grew pines in these soils. So I had this kind of factorial set of treatments, so these two different soil origins. And then I manipulated the arrival time of ectomycorrhizal fungi. So I collected spores from a species of ectomycorrhizal fungi, this Suillus pungens thing. And I added those spores to pots that these seedlings were growing in at different times, either at the beginning of the experiment, after 1 month, after 5 months, or never. And then on top of this, I also grew the pine seedlings by themselves or in competition. And I grew them in competition with this sort of dominant scrub species, Baccharis pileolaris, that you can see kind of dominating the foreground of this picture. So I'm going to show you just visually kind of what the results looked like. And I will say I'm only going to show you the data right here from these far soils, okay? And the reason for this is that the soils that were close to the edge of the forest had tons of ectomycorrhizal inoculum in them. All the seedlings were colonized, and none of the inoculation treatments did anything interesting. They just acted exactly like these early inoculation treatments. But in the soils that were collected from far away from pines, what you can see is that depending on when I inoculated the ectomycorrhizal fungal spores into these treatments, had a really big effect on the growth of these plants. And so if you inoculate the plants early on, the pine seedlings do really well. They're quite large. And as you wait progressively longer and longer time periods, the plants get smaller and smaller. Okay? And in competition, what this does is it actually, you can't really see this very much, but these little guys right here, these are all little shoots of the Baccharis growing up, and the Baccharis actually does quite a bit better as you delay arrival of these ectomycorrhizal spores. So one of the neat things to me that you can do with this data set now is actually look, by taking all the data and putting it together, you can look interactively at the effects of competition and mutualism on the growth of these pine seedlings. So on the y-axis here, I'm showing you the total biomass of these different of these pine seedlings, and the green bars show the pines when they're growing by themselves. So this right here. These orange bars show the pine when it's growing in competition with the Baccharis, and these are organized by these different inoculation treatments. So these are seedlings that got spores after 10 days, spores after 41 days, after 144 days, or never got spores here. And what you can see just visually is that there is a significant negative impact of having these competing plant species around on the pines. So they tend to be about half as large when they're growing in competition with this Baccharis. But for me, what's actually quite striking is if you compare across these sort of early inoculation and late inoculation treatments, the mutualist effect is actually quite a bit larger than this competition effect. The decrease in biomass from having spores early versus late is about a 5-fold decrease in biomass. All right, and so from this you can actually conclude that in this particular system, competition is actually a relatively smaller force than the importance of these kind of positive interactions or mutualism. One thing that was actually a little bit surprising to me about this particular data was that I had thought that the strength of competition was going to be a lot greater in these treatments here where the plants never got their mutualists. And in fact, I found that in this particular graph that there's really no interaction between these two treatments. Competition seems to have sort of a constant negative effect on relative growth rate, but the absolute relative growth rate seems to get set by the presence of these ectomycorrhizal fungi. And in fact, this actually only shows up if you log transform the data. If you don't log transform the data, you can't actually even detect the effect of competition in these smaller seedlings because they're so small. There's actually not much biomass to be lost. And in fact, competition looks worse when the ectomycorrhizal fungi are present because there's actually a larger absolute fraction of biomass that's lost. All right. So one of the things that I think is actually quite interesting about this is I think that these ruderal ectomycorrhizal fungi may actually act as keystone species in this particular system. And I say that because when these are the sort of key fungi that colonize these pines, and they seem to be able to determine the outcome of competition between pines and other coexisting vegetation. And we know in this system that after fire there's quite a bit of vegetation shifts in the dominant vegetation that happen. This is actually Point Reyes, and this is that fire in 1995. You can see it was kind of a moonscape. And this was a study that was actually done by my wife. And she looked at these different pre-fire and post-fire vegetation vegetation cover. And what she found is actually quite a bit of transition, say, from coastal scrub to bishop pine or coastal scrub to ceanothus type vegetation. All right. And if the presence of these pines is really important, or these ectomycorrhizal fungi is important to determining who wins, then there are some interesting ramifications for that. Because we've gone out to these different types of vegetation at Point Reyes. So these kind of three dominant vegetation types: Baccharis, ceanothus, and pine. And this is a postdoc of mine, Marie Duhamel, who crawled around out there. And if you've ever done work in tropical rainforests, working in the coastal scrub is way worse and way harder than working in a tropical rainforest. You're on your hands and knees a lot more than you would be in a tropical forest. And so what we did is actually take advantage of this the sort of pre-fire vegetation map. And we went to 30 places where we knew the pre-fire vegetation type and knew the post-fire, what it had become after fire. And so we looked at 10 independent transitions from, say, Baccharis to Baccharis, Baccharis to ceanothus, or Baccharis to pines. And one thing you'll note from these is that all of these different species have different below-ground microbial symbioses that they associate with. And what we found is that as you get these transitions between vegetation types, you get what I would almost describe as alternative stable states in the soil. So certainly from the perspective of the microbes are quite different, and these things persist for multiple microbial generations. So you could accurately describe these as alternative stable states. And then the properties of these ecosystems are actually quite different too. This is a PCA showing kind of soil chemistry data from these different sites. And you can see that the three different veg types split out pretty nicely. And this is based on things you might expect. So ceanothus is an end fixer, so you can see that nitrogen concentrations in the soil are pretty high underneath the ceanothus, whereas you get differences in litter carbon concentration and the rates of decomposition as you go to these different vegetation types. Okay? And so if the fungi are actually facilitating these shifts between different ecosystem states, these kind of local scale interactions actually might ramify up to influence landscape scale patterns of vegetation. So this is basically what I was going to say here in the summary slide. What I've shown you here is that we get this landscape scale heterogeneity in the ectomycorrhizal community, and hopefully what I've shown you is that this landscape scale heterogeneity in the ectomycorrhizal distribution of ectomycorrhizal fungi can then influence these local scale plant processes, or local scale competition outcomes. And then this may actually cascade up to then change the kind of pattern of vegetation we see across a given landscape. And I think the two important things to also take away from this particular part of the talk are that, for me, it demonstrates the importance of mutualisms. They can actually, in some cases, be more important than the negative interactions we frequently think about. And the second thing that's important to me is that because the timing of arrival of these ectomycorrhizal fungi was important, it suggests that mutualist limitation may be more important or more common in these for plants than we might previously expect otherwise. Okay, so what I want to do now is shift gears and sort of move even higher up in spatial scale. And I want to start to talk to you about some of the work we've done trying to look at continental scale patterns in the biogeography of soil fungi. And for the reasons I explained earlier, this idea that microbes were essentially dispersal unlimited, people for a long time had assumed that microbes wouldn't show any meaningful biogeographic patterns. And just as evidence of this, if you look at the names of fungi that have been applied in places like California, basically all of the mushroom species in California have European names. This is because when people got here, they thought, oh, these are probably just the same things we've seen at home, because these things are all over the place. So we really wanted to try and get at this and try and see whether or not there actually were kind of normal-looking biogeographic patterns for these kinds of microbial organisms. So we were lucky enough to get funding through this cool program that the NSF had called the Dimensions of Biodiversity. And for this, we were trying to look at sort of taxonomic and functional diversity of soil fungi across North America. And I'll say this is a collaborative project between myself, Tom Bruns, and John Taylor at UC Berkeley, and Reyes Vilgelis at Duke University. And what we did is go all across North America and collect huge amounts of soil. And you can see that this is a map of North America. These black points are individual sites that we'd gone to. So we had 71 total sites. We collected something like 1,500 soil cores, ranging all the way from Florida up into Alaska. And we extracted DNA, sequenced the fungi that were present in these soils using next-gen sequencing techniques like 454 and MiSeq. Generated a lot of fungal DNA sequences in the process. One thing I'll note that I should have said earlier is we tried to restrict our sampling to vegetation that was dominated by pine species. And this is so we could remove any potential artifacts that might be introduced by host preferences between mycorrhizal fungi, because we know that these things exist, but usually only at the family level. So we thought we could control for this somewhat. So just sort of some like bird's eye view kind of results. We found that the soil communities of fungi were really dominated by these ectomycorrhizal fungi. They were most of the sequences we collected, which is just some indication of how important these kinds of mutualisms are for structuring soil microbial communities. Kind of median alpha diversity, this is ectomycorrhizal and saprotrophic fungi, was about close to 200 species per sample. And these samples are about the size of a baseball. So this is a fairly high level of alpha diversity. And across all of our samples, so just from these small sampling of soils, across this one particular vegetation type, we detected about 16,000 species of fungi. And just to put that in perspective, there were some recent estimates that the entire Amazon basin has 16,000 species of trees, and the entire North American flora has just about 15,000 species of vascular plants. So fungal alpha diversity and beta diversity are actually quite high. So I was really interested in this question of whether or not the biogeography of these organisms was really just driven by the environment, whether or not everything that we would see at this larger scale was really just environmentally driven. And so the way that I wanted to try and get at this, and this was the expectation you'd have under this kind of Bass-Becking view of the world where everything is everywhere, but the environment selects. So I want to try and get at this question is by showing you an NMDS ordination. And I'm kind of proud that I've gotten through most of the way through a microbial ecology talk, and I will only show you one NMDS ordination, which is, I feel like, is somewhat of an accomplishment for me. And what I will show you is each of these individual circles represents one of the soil cores that we took. And the closer together these points are in space, the more similar these fungal communities are to each other. And so these are quite similar, these are quite far away. Quite different. And what I will do is color code these, or code these, depending on where in the soil horizon I took them, either from the upper kind of organic soil or the lower mineral soil. That's the kind of small versus large dots. And they're going to be color coded depending on where in North America they came from. And the reason I've chosen to do it this way is we found that in this sampling that soil horizon was really the predominant factor that determined the soil chemistry, the chemistry of the soils that we sampled. All right, so by sampling across this gradient, we could actually get soils that were in really different places but had very similar kind of chemical compositions relative to each other. All right, so if we end up seeing an NMDS ordination that looks like this, where all the points cluster based on the size, what you're seeing is like strong environmental selection on these microbial communities. All right, this is similar, this would be consistent with this kind of Bass-Becking view of the world. But if you see clustering that is basically mostly determined by color, this is suggesting there's a strong geographic signal. And this is much more consistent with kind of biogeographic patterns and processes we've seen with other types of organisms. So you can probably guess what it's going to look like, but to me this is actually quite nice to see. You see really strong and geographic clustering of these different soil fungal communities. So you can see these are our East Coast fungal communities over here, these kind of boreal Alaskan ones here, and a lot of our West Coast ones over here. And you really can't pick out any signal for soil horizon, even though that was the dominant thing that predicted soil chemistry in the system. So I won't show you any of the sort of statistical analyses we did, but we show that climate does play some role in determining the sort of structure of these fungal communities. We also find a really important role for kind of pure space, which is consistent with dispersal limitation as being one of the things that drives the patterns that we end up seeing here. Interestingly, when we look at the function of these communities, and so this is function measured by, we measured, I think, 9 different enzymes that are associated with a turnover of things like carbon, nitrogen, and phosphorus. You see a very different pattern. So these are principal components of each of these different soil enzymes. You can see that there's really no geographic pattern in the sort of functioning of these communities. Rather, the functioning of the community seems to be explained primarily by the local soil environment. So this is a PCA of the soil chemistry, which is primarily loaded on carbon and nitrogen in the soil. And then this is this soil enzyme. And you can see all the small points for the organic horizon are up here. All the kind of large points for the mineral horizon are down here. And this is a fairly decent relationship. What's interesting is you see these points that are pretty far away from each other geographically, but pretty close to each other in their functioning. What this suggests is there's functional convergence across these different communities, and by extension that there's quite a bit of functional redundancy probably in these diverse soil communities. So one of the fun things we've also started to play around with this kind of data is taking it and trying to see if we can come up with models that predict larger scale patterns of ectomycorrhizal diversity and map this across the continent. And so this is a map. We find that climate does a pretty good job of predicting ectomycorrhizal richness at the site level. And so this is our kind of map of predicted ectomycorrhizal richness across North America, masked by places where we expect pines to be growing, so not really down in the Great Plains. And one of the cool things that comes out of this is that these relationships between climate and mycorrhizal diversity are actually quite nonlinear. And there's actually multiple kind of multiple peaks of richness. So you can see down here in the southern United States, we get a kind of a richness, a hot, wet richness peak. And over here, kind of in the northwest, we get this kind of cool, dry richness peak. So we've done, as one would do with this kind of a map, taken future climate scenarios and tried to predict how ectomycorrhizal diversity is going to change in the future. And because of the multiple modes for diversity and because of non-linearity, we get fairly complex predictions across North America, where you see places like the southeastern US are actually going to gain quite a few species, where you see a lot of loss of diversity in the southern boreal forests and gains in the north. All right. All right. So what I would say from this is that, just to sort of summarize this part of the talk, we see at the continental scale patterns of biogeography that are consistent with other kinds of macroorganisms. And our data suggests that a lot of this diversity is probably somewhat functionally redundant, at least from the perspective of nutrient cycling. And I think establishing these kinds of patterns are actually quite important to make sort of microbial ecology a bit more of a predictive science. And so if you asked me to predict what kind of fungi would be in a particular sample, what I would ask you first is where in the world did that sample come from? That'd be the first kind of piece of information I wanted to know. If you asked me what those organisms were doing, I would want to ask you something about what the chemical concentration of these soils would look like. And people have actually used this, this kind of information now to begin to build like forensic models for microbial communities. And so you can actually use the fungi in particular in a sample, some sort of forensic sample, to determine where in the world this sample came from, just to give you some illustration of how powerful these things can be. And so while I didn't show you any direct evidence that dispersal limitation was driving these patterns, we have a lot of evidence that's consistent with these sort of same kinds of dispersal limitation and the importance of dispersal barriers that we see at the landscape scale, that these things actually scale up to help generate some of these unique mycofloras that we observe in different parts of North America. And I should say that other studies have come out that seem to kind of support the kinds of things that we've shown here. All right. And the direction that we're really going in our lab is to try and take some of these large-scale patterns that we've observed in terms of continental variation and the structure of these ectomycorrhizal communities and the climatic drivers we think are behind those, and then try and understand how that is going to then influence things like the prevalence of particular types of mycorrhizal symbioses, how these plants might then compete with each other in different climates, and how that's going to structure vegetation across a landscape. Okay, so just to summarize what I've said here, if there are any conclusions, there's one conclusion I hope that you walk away from this talk with. It's that while there has been this sort of prevailing view of microbes as being dispersal and limited, I think there's a preponderance of evidence that suggests that that really just isn't the case. That dispersal limitation is really important, both at local scales for driving the local kind of community dynamics of these microbes, and also at much larger scales for the evolution of regionally endemic microbial communities. To me, the fact that dispersal drives so much of this kind of spatial variability actually makes microbial communities much more interesting. Because if microbes are really ubiquitous and they were dispersal and limited, no one would need to think about them, right? If you were a plant, you just have the right or the wrong microbes, they'd be present, and you wouldn't really need to think about the dynamics of these organisms. But because you get so much spatial variability, you need to understand the community structure of these microbes in order to understand other things like ecosystem cycling or plant community dynamics. So the other kind of takeaway I hope you get here is that mutualisms are actually more important perhaps than we've given them credibility for, and that they're probably equally as important in many cases as the more traditionally studied antagonistic interactions in ecology like competition and predation. All right, and finally, I just hope, and you guys are in the middle of searching for a microbial ecologist, so you probably understand this, but that we think that if most of the diversity on this planet is microbial, most of it's bacteria, viruses, and fungi, we now only have the tools that we now only have access to the tools that enable us to rigorously study the ecology of these communities, and this gives us this great opportunity to test in new systems whether or not the ecological theory we've mostly developed with plants and animals actually works when you apply it to these different types of organisms. And if it does work, that's great positive confirmation of the value of these theories. And if it doesn't work, then it means we probably need to develop new ecological theories. So I hope that next time you look across this sort of a landscape, you sort of see it from the perspective of the microbes and think of them not just as passive kind of indicators of whatever is there, but also think about the active role that they play in shaping the patterns of vegetation that we see across these kinds of landscapes. I just want to acknowledge kind of funding agencies. I've been funded by the National Science Foundation and the Department of Energy, and all the wonderful people in my lab that have generated a lot of this work and made being a professor an enjoyable process. And I'd be happy to answer any questions. Yeah.
Thank you. Very interesting talk. I really enjoy.
Great.
My question is actually, is there any association between nutrient poor soil and fungi?
Between nutrient pulses.
Like always we talk about so many research going on, Cape Town, Southwest Australia, or here in our shop. I was wondering if you would have a control experiment, low nutrient next to each other, fungi would, you would find more diversity in nutrient poor soil?
Yeah, so that's a great question. I don't know of anyone, and this is the kind of thing that probably is more and more possible. There have been some, I would say that probably the easiest way to answer this is that humans have done a lot of natural experiments where we increase things like the amount of nitrogen that is being deposited in soils. And generally what people have found is that as you deposit, say, more nitrogen in an environment, you tend to get a decrease in the diversity of these ectomycorrhizal fungi. And the reasons for this kind of economically make sense from a plant perspective. If someone else is supplying, if the environment is supplying the resource that you critically need, there's no reason to keep trading with these mycorrhizal partners anymore, and so oftentimes the plants will drop these mycorrhizal fungi. You can do this in pot experiments as well. If you just fertilize a plant, it can become non-mycorrhizal pretty quickly. Yeah.
So we can say that there's a coevolution?
Absolutely, absolutely.
Between nutrient poor.
Yeah, there certainly is. And one of the things I should say is that I didn't really get into the contrasting the physiology of ectomycorrhizal versus arbuscular mycorrhizal vegetation. One of the ideas is that the reason that ectomycorrhizal symbiosis evolved was in kind of colder, more temperate environments where decomposition rates were slower and more of the resources are tied up in organic matter rather than mineral nutrients. And so plants actually needed to compete with microbes for these kind of limiting resources. And so they evolved a symbiosis with the fungi that are more closely evolved from saprotrophic fungi that have higher capacities to degrade organic matter and take it up. And so oftentimes people expect that in systems where N is the limiting nutrient, you often see a dominance of ectomycorrhizal symbiosis. And where phosphorus is the limiting nutrient, you often see a predominance of arbuscular mycorrhizal symbiosis. That might be an oversimplification of reality, but that's kind of the paradigm that most people think of. Yeah.
I have two questions.
Yes.
The first concerns the study at Point Reyes.
Yeah.
You didn't mention anything about the mechanism of dispersal. And so it's important because if dispersal is by air, then wind direction plays a big role.
Yep.
If you're downwind, you're more likely to get dispersal. And if it is by water, then being downhill is more important.
Yeah.
And so what about that?
So I think dispersal is probably a somewhat complicated process for these guys, and I should say that mostly the vast majority of fungi have aerially dispersed spores, so they're probably mostly going to be moving through the air. That doesn't mean that they're not going to be moved in the soil through groundwater flow if they do get washed out into the soil, although it's unclear how far they actually move through the soil matrix or if they just sort of stick once they get there. There are fungi that have other dispersal mechanisms. So truffles, or if anyone has a sort of expensive taste for food, truffles are animal dispersed, and so they get moved around and scat, and that happens with a lot of the fungi at Point Reyes as well. So they're mostly aerial dispersed. I think they get washed out oftentimes when it rains. And I thought a lot about wind direction in these studies, and I don't know if I can go back to one of these slides. The prevailing wind, sorry, let me get there. Is this where I wanted to go? Yes. So the prevailing wind in this part of the world is probably from the northwest, so it's coming in this way. So I did a lot of trying to think about how wind direction was going to influence these things, but I couldn't really find a really great way to measure it, and it didn't seem like the patterns I saw were consistent with kind of higher colonization downwind. It seemed like the strongest signal was distance in this direction, or distance anywhere you were away from where these pines were. But I wish if I had had more money and resources, taking a more sophisticated approach to trying to understand the sort of dispersal patterns might have explained some of the extra noise in my data set. Yeah.
Okay. The second question concerned your made remarks about species diversity of microbes versus various animal groups or plant groups.
Yeah.
I don't have an understanding of how you determine using molecular data alone what constitutes a species.
Yeah.
And if you applied those same techniques to macroorganisms and ignore the alpha taxonomy, and you compared them, would you still get roughly the same results?
Yeah, so it's a good question. I think it's going to be so variable depending on which taxonomic group and which locus people are using. What I would say is that for fungi, using this internal transcribed spacer region, one of the nice things about fungi compared to, say, bacteria is that there's actually a reasonable species concept for many, at least of the fungi that produce macroscopic fruiting structures. So we think we have an idea of what a species might be. And we can compare these molecular markers to our kind of normal species concept. In the case of fungi, we think that this marker we use and a 97% sequence similarity cutoff actually approximates a fungal species pretty closely using kind of normal taxonomic methods. I don't know how that would translate to plants. Again, it's going to depend a lot on what the marker is, how conserved it is. But I would say that this is a really important point, because a lot of times people will take microbes like bacteria or some fungi using particular genetic markers that are highly conserved, and they'll look at global scale patterns in biogeography and diversity, and they'll see a total lack of any pattern. Looks like everything is everywhere. But when you're looking at it, a lot of these markers, they're probably more akin to something that you would think of as like family level for plants. And if you start to look at the family level distribution of plants, that also looks like everything's pretty much everywhere, right? And there's a lot of things that are pretty broadly distributed. And so I think some of the arguments about whether or not everything is everywhere are really hung up on inconsistent definitions of what is the thing that we're thinking about. And someone recently did a study where they published a paper in Science saying that arbuscular mycorrhizal fungi were cosmopolitan. They were really on every continent. There was no variability between places. And one of my old advisors took the same locus they used, a different one than we normally use for this group of fungi, and he applied it to animals, and he found that if you use the same locus, you would actually lump humans together with wombats into sort of one big OTU. And so I do think it is important to get the right sort of taxonomic resolution when asking these questions.
I think we should stop there.
Okay, great.
Chance to come up, but please join me in thanking Kabir for a great talk.
Yeah.